We like the scent of humid soil after a rain shower. What we smell is geosmin, a volatile produced by bacteria living in the soil. At the Netherlands Institute of Ecology, we learn from nature. Our research group studies the role of volatile organic compounds in microbial interactions in the soil. Microorganisms such as fungi and bacteria produce enormous amounts of volatile organic compounds, or volatiles for short. Volatiles have unique properties that enable them to diffuse from the liquid or solids to the gas phase. In soil and the rhizosphere, the soil attached to plant roots, volatiles can travel over long distances through water and gas-filled pores. That's why they play an important role in long-distance communication and competition between soil microorganisms. For a long time, microbial volatiles were overlooked in ecological research. But with the advance of detection techniques in the last few years, research on microbial volatiles has increased in the medicinal, pharmaceutical, agricultural and ecological fields. At the Netherlands Institute of Ecology, we have developed a versatile method for analyzing a wide range of microbial volatiles. To collect volatiles, we have designed glass petri dishes with special lids that have traps fixed to them. After heating, the volatiles are analyzed on a gas chromatography mass spectrometer. With this new technology, we're able to look deeper into the role of volatiles in microbial interactions.
Volatiles act as infochemicals between microorganisms. Most studies have investigated the effect of bacterial volatiles on fungi, but not on how fungal volatiles affect bacteria. In our study, we investigated how fungal volatiles affect bacterial behavior, like for example their growth, their antimicrobial activity, their motility or their biofilm formation. And we investigated how this response was dependent on the fungal substrate composition and on the growth stage. We selected different soil bacteria and fungi, and we exposed bacteria to fungal volatiles by using a two-compartment petri dish system. Volatiles emitted by different fungi lead to changes in motility in especially two bacteria. And these changes in motility were dependent on the fungi's substrate composition and growth stage. Some of these fungi produce a very specific cluster of volatiles, like for example terpenes. We confirmed that these terpenes cause the motility changes in these two bacteria.
So far, most of the studies on bacterial volatiles were performed on bacteria cultivated in monocultures. But we don't know how bacterial interspecific interactions influence the activity and composition of volatiles against fungal, oomocetal and bacterial model organisms. In our study, we selected phylogenetically different bacteria and determined the identity and antimicrobial activity of their volatiles produced in monocultures and in pairwise combinations. We see actually that bacterial interactions influenced the volatile blend composition, and we also saw an effect on volatile compound production. For example, we saw an induction or an silencing of certain volatile organic compounds, like for example indole. When exposing fungi and oomocetes to bacterial volatiles, we see that their growth is actually reduced. The effect of volatiles on bacteria varied between no effect, growth inhibition to growth promotion, depending on the volatile blend composition.
There are many experiments on volatiles where microbes are incubated on agar blades. but to understand their actual role of volatiles and microbial interactions in soil, we thought to move away from agar blades to a more natural system. We designed a system where we have soil mixed with the microbial community and root exudates that mimics the rhizosphere. With the system, we could see that the volatile emission is affected by microbial interactions, but also by shifts in the microbial community composition. In a follow-up experiment, we looked at the actual role of those volatiles produced by microbial interactions in soil. So we exposed microbes not having access to root exudates to volatiles of microbes having access to the root exudates. In this experiment, we could see that the activity of those microbes was strongly increased. Therefore, we could show that volatiles produced by interactions of microbes in the rhizosphere have a long-distance effect on microbes in the nutrient-depleted surrounding soil.
Together, we aim to better understand the ecological role of volatiles in microbial interactions in the soil and the rhizosphere. Studying the volatile production of interacting microbes might lead to the discovery of new agrochemicals that help to reduce the use of pesticides, and antimicrobials that can combat multi-drug resistant pathogens.